Hi guys, welcome to my biocast. In these videos, I cover some of the most talked about stories in biotech over the past month, and then I tell you what I think about them too. But as well as chucking my thoughts on top of them, I'm also really interested to know what you guys think about the story I'm going to cover today or any of the stories that I've covered in my biocast. So let me know what you're thinking in the comments below and we can start a conversation. And the story I'm going to cover today is the arrival of AI in the drug discovery process. Atomwise and Enamine have launched an AI-powered virtual drug screening program built on Enamine's virtual library of 10 million compounds and Atomwise's deep learning neural network AtomNet. Using this platform, Atomwise can evaluate the binding of billions of compounds to cancer target proteins, with the focus on small molecule drug discovery for pediatric cancers. Deep learning neural networks are used every day for things like autonomous cars, speech recognition, and advanced image recognition computer vision. The tech works by breaking down complex concepts into small fragments, and then learning what combinations of these fragments fit to make the original concept.
One of the interesting characteristics of deep learning is the fact that it can take very complex concepts and compose them out of smaller and smaller and smaller concepts to capture something very complicated. So for instance, if I were to ask you to write a series of rules that define what a face is, you could start trying to write them all out, but it gets pretty complicated very fast. So in this instance, what you do is create a neural network that has many layers, and we might start with raw pixels of an image, compose those pixels into patches that form edges, like diagonal and vertical edges as you see here, and then compose those edges into pieces of faces, for example, like noses and mouths and eyes, and then finally, in the final frame, assemble all of those together to form a concept of a face. And so the question that we asked ourselves about 3 years ago when we started doing this work was, could we do the same thing, but for chemistry, organic chemistry, and for drug discovery? So the same technology that differentiates if an image is a dog or a cat, or a waveform is a cat or a car, could we look at a molecule in a binding pocket on a protein and say, is this a binder or a non-binder?
In early June, AtomNet signed a multi-year deal with Eli Lilly and Company to use AtomNet to support Lilly's preclinical drug discovery operations for 10 compounds. Atomwise could receive up to $1 million per target in discovery milestones and up to $550 million in commercial milestones. They also have the option to take forward compounds from the collaboration that Lilly choose not to use. Other big names that Atomwise has bagged partnership deals with include Charles River, AbbVie, Merck, Duke University School of Medicine, Bayer, and the Karolinska Institute. Abraham Heifetz, CEO of Atomwise, says, "Many of our partners have successfully identified early drug candidates, including sub-micromolar hits, by screening only 10 million compounds with our AI virtual screening platform. We've barely scratched the surface of what is possible. Imagine what will be found when we screen a chemical library that is 1,000 times larger." Other AI platforms are also beginning to infiltrate big pharma, with Benevolent AI signing a long-term collaboration deal with AstraZeneca in May to use machine learning to develop new drugs for chronic kidney disease and idiopathic pulmonary fibrosis, alongside signing an exclusive license deal in 2016 for several clinical stage drug candidates with J&J's Janssen Pharmaceuticals. Pfizer and Bristol Myers Squibb, meanwhile, have enrolled Concerto Health AI to focus on AI for oncology-specific real-world data and real-world evidence generation. Smaller biotechs are also seeing the potential of AI and machine learning, with Oxford Biomedica signing a 2-year R&D collaboration with Microsoft Research to improve the yield and quality of next-generation gene therapy vectors. And whilst the promise of shiny new tech crunching hundreds of molecules to find the perfect candidate is enticing, we might need to slow down before putting all our faith in silico methods of discovery. In a critical review of a paper by Thara Karaman et al. Describing the de novo design of antibodies to neutralize the H7N9 strain of influenza and the Zika virus, authors William Finley and Alexei Lugovskoy say, "Every new antibody engineering technique had required years of optimization before robust, reliable processes were attained. The antibody engineering field has learned multiple painful and costly lessons from early experiences with mouse monoclonal antibodies that were immunogenic in man, antibody humanization techniques that led to the loss of affinity where iterative protein engineering is needed to restore function. It is difficult to believe that the huge leaps forward in de novo antibody design that will de-risk drug discovery could be achieved without iterative learning on how to address such issues. If antibodies are ever to be truly designed fully in silico, it is essential that they are optimized for all characteristics of a successful antibody drug, and not only for high complementarity to their target antigen of interest. So this story has been fascinating for me and awakes a sort of conflict that I have between loving huge technological advances and having a very founded and real fear, I think, of machines taking over. But with regards to this story, I think the incorporation of AI platforms in drug discovery are well overdue, especially compared to other industries like aerospace, where they use simulations to model and test new aircraft designs. But I do agree with the critics in that we should understand more about how this technology works and where it should be applied, like in the case of the human germline editing that I covered in a previous biocast. You can click there to go watch. It may be hugely beneficial at the start of the drug discovery process for small molecules and will save huge amounts of time and money that is currently spent on wet lab techniques at this stage. But testing those candidates prior to preclinical testing is still important to verify and select the most promising candidates in a physical system. On the flip side, for biologics, we still don't know exactly how antibodies interact with the immune system, so trying to synthesize them from scratch for clinical use is, in my opinion, really premature. But again, AI and machine learning could be hugely beneficial as an initial screen for target specificity and binding for these molecules. So that's the story, that's my opinion, but what are your thoughts on AI and machine learning in the drug discovery process? Should we be exercising caution as we unroll this tech into the field? Or should we just jump in and let the machines loose so that they can learn and answer the problems that we are stuck on? It's an exciting field, an exciting time in biotech, and I would really love to know what you're thinking. So guess what? If you've made it this far, you've made it to the end of this video. Congratulations, and thank you so much for watching. And whilst you're here, you might as well go and check out our antibody analytics company page. We put out loads of information about the types of assays that we're working on, as well as the latest biotech news that I don't cover in my biocasts. And also memes and videos and the stuff that we do because it's 2019. Why not? So once again, thank you so much for watching. I hope you enjoyed the video and I'll see you on my next biocast.